Hello, good afternoon, everyone. I'm very pleased to be there to present Coave Therapeutics, and thank you for making the time. I know there's a challenging time with unfair competitions from Pata Negra and other tortillas upstairs, but happy to have you on board. So Coave Therapeutics, we are a genetic medicine company developing a new delivery technology with first applications in CNS and ocular space. So like many others, we try to generate a new class of vectors to address the limitations of the current available technologies in terms of efficiency and safety. What we do is we combine the best of different technologies. From the viral side, the AAVs, and on the non-viral side, the conjugation technologies that have been already applied to many different other products, from antibodies to nucleic acid. So what we do, we functionalize AAV by using a specific conjugation technology and binding onto those AAV specific rationally designed ligand. And our strategy is to target key limitations such as tissue tropism, transduction efficacy, liver detargeting, and also the immunogenicity of capsid. So the company is based on this platform called Alligator for AAV ligand conjugate. From this platform, we have already generated few therapeutic programs in the CNS space, and we are also engaging the platform with third party. We have 3 ongoing collaborations with companies US-based for evaluating our platform. So the platform, Alligator, so it's a very differentiated approach compared to other platforms currently developed. We are not doing directed evolution. We are not changing at all the AAV amino acid compositions. We have developed this technology from day one to have a one-step chemistry downstream the AAV production without, again, introducing unnatural amino acids. So we work with different AAV serotype. We have been able to work with third-party product from CRO product from our internal AAV product, and our technology is around the design of the ligands, the specific linkers that we are using to bind specifically on lysine residue at the surface of the capsid. So basically, the way we work is we design the ligands based on knowledge and actually human biology, so we don't generate millions of ligands. We have a very limited libraries of few dozen of them, and then we can directly test our conjugated AAV in vivo models without the need to do enrichment cycle and selection. So what we have seen is we have a very robust and consistent translations of the improved property from rodent to primate. And very importantly, this is the only technology that enabled to modify the capsid of the existing therapeutic product. So with our ongoing partnership, we are directly modifying AV9, AV8 base product without changing the whole capsid. So we are not changing the plasmid design, the USP and DSP developed. We can directly work on existing AAV capsid. So we have initially focused our work over the last 3 years on small molecule ligands that were developed with a specific small molecule end group. So we work with a family of molecules that have been selected because we were targeting certain receptors on neurons and photoreceptors. Now we are moving to a new class of ligands where instead of having a small molecule, we are using peptides and aptamers as an end group. And with this new ligand technology, we will be able to address different tissues, different route of administrations. This is ongoing work. We expect to have our first demonstration of in vivo efficacy of those peptide/aptamer-based ligands in the coming month. And one of our primary interests this year is to have an IV BBB penetrant capsid for CNS delivery. So how the conjugation impact the biology of the AAV capsid. So it's a major modifications. We can have up to 700 ligands to the surface of the capsid, which means that we interact a lot, or we change a lot the interaction between the capsid and different components. What we have seen in vivo, again in rodent and in primate, in two different tissues, the retina and the brain, is we change the biodistribution and the extracellular navigation of the capsid. We have, of course, seen that we improve the targeting because we are creating a secondary receptor interactions with the ligands and the targeted cells. And very clearly, over the last month, we have seen that our vectors and the conjugation impact the intracellular trafficking. And probably the reason is linked to where we bind those ligands on specific lysine residue that are involved in the natural trafficking of AAV. On bottom line, what we see, again, on different tissues, we have a very more rapid and higher level of transductions and mRNA expressions per VG compared to a traditional serotype. So we have initially worked on conjugated AAV for improving AAV2 for local administrations in both the brain and the retina. And in these studies, we have demonstrated we improve the transductions of AAV2. We make even conjugated AAV2 even more efficient than AAV5, for instance. We have demonstrated we have this very nice spreading of the capsid from the site of administration. And what is very well known with AAV2 and others is when you inject directly in the tissue, you have those capsids being trapped on the site of administration. Then, over the last 18 months, we have been working on conjugated AAV2 and conjugated AAV9 for what we call fluid compartments, so CSF administration in the brain and IVT administration in the eye. And here now we have fresh data on primate demonstrating we improve the transductions with the conjugations and we reduce the off-target transduction. And finally, and this is what we are developing this year, as we have seen that the capsid could be detargeted for the liver, as we have additional new data confirming that we have less blocking antibody interacting with our conjugated capsid, we think we are taking the right properties to enter the IV administrations. And as I said, we are developing BBB penetrant capsid using our conjugation technology. So from the initial small molecule ligand library, we have been able to modify 6 different serotypes. We are working on others. Doesn't mean that it doesn't work on other serotypes. It's just a matter of bandwidth and resources. But as you can see on this slide, we have been able to modify all the classical serotypes, even second-generation serotypes such as 7m8 have been modified with our technology. In terms of validation, first, for the intraparenchymal, intraputamen route of administrations with the conjugated AAV2, what we have demonstrated, this is our best capsid, this conjugated Coave that have been initially tested versus AAV2 in rodent. And what you see on this slide is from the site of administration, which is in the striatum, we have a very nice pattern of distribution and very more intense expression of the protein from the striatum up to the substantia nigra. And in primate, on the right side of the slide, we have seen the same. Basically, from the striatum, we have been able to cover up to 80% of this structure with our conjugated AAV2, and the dose which was used in this setting was 7.710 to the 10 VG, which is a very low dose for this route of administrations. And same, we have seen, like for the mice, an expression, strong expression is the substantia nigra. And we did this in comparison to AV2 and AV5. And basically, we have improved by twofold the volume of structure covered with our conjugated AV2. At the same time, we improve the transduction. At the same time, we improve the biodistribution. We do not change the cell tropism. So the conjugated AV2 are infecting neurons like AV2 and not other cell type. So you will see later on that, no, you won't see, but what I can say is on the primate, with this capsid, we have now validated with a therapeutic transgene the same pattern of distribution. For our program for GBA1 Parkinson, we have confirmed that in primate, from the putamen administration, we have a strong expression of the GBA1 gene and GCase activity in the putamen, in the substantia nigra, globus pallidus, and caudate, which are the key brain structures to be targeted in this indication. So it's an additional confirmation on top of the GFP data presented here. So we recently also ran another studies, again in primate, for the intracisterna magna route of administration. So it's an administration in the CSF at the back of the brain. And the limitation currently with this route of administrations, when often AAV9 is used as a vector, you have a very limited distribution to the brain, almost no distribution in the deep structure. A lot of AAV9 is actually transducing DRG and going to the liver. We did this multiplex study where we have co-administrated conjugated AV2, conjugated AV9, AV2, and AV9. So what you have here is the level of mRNA. So we have been focusing a lot on what we think the most relevant parameter, which is the mRNA of the transgene expression, to AV9. So again, it's a true comparison. AV9 was co-administrated at the same dose than the two products here. So what we see is this conjugated AV2 candidate has a clear superiority in transduction for different parts of the brain, of the cortex, as well as the cerebellum and the midbrain and substantia nigra, compared to AV9, up to 100 times increase in terms of transgene expression. At the same time, we have a clear superiority to AV9. We still have a very low to almost no transductions of our target tissue. So we have a reductions of DRG transduction by more than 10-fold compared to AV9, and a reduction of the liver transductions that more than 100 times, actually 700 times for the liver. Those scales are logarithmic. For the conjugated AV9, we confirmed that we have a good CNS transductions for the conjugation, but very interestingly, we have a reduced as well transduction of the conjugated AV9 in both the liver and DRG, which is, again, one of the key limitations. So we are very pleased and satisfied with these studies, and we will confirm in the next study those two lead capsid for ICM administration. So in terms of pipeline, as I mentioned, we have actually 3 programs ongoing using the conjugated AAV. The first one is for GBA1 Parkinson. In this one, using a conjugated AAV2, we have demonstrated in a GBA1 deficiency mice model that we restore the GCase on a dose-dependent level. We have also demonstrated, as I mentioned before, that we've confirmed the nice pattern of distribution of the GCase in the deep structure of the brain, which is a key one. This program is progressing to IND-enabling study. We are preparing the GLP tox. We are targeting actually IND filing early 25 for this program and enrolling first patient by the end of 25. We have 2 other programs using another transgene called TFEB, which is an activator of the autophagy pathway. Those programs are in preclinical stage for a MSA, multiple system atrophy, and ALS. And finally, our historical program using a classical AV5 for a PD-L6 beta retinitis pigmentosa. We have finalized the phase 1/2. We are currently reading out the 1-year data set, and we will know very soon the outcome of this 1-year follow-up. We have a 2-year follow-up plan also next year. This program has been already partnered for the European right only with TEA, which is an ophthalmology company in Europe. We still have the ex-Europe right, and we are currently investigating potentially licensing out this program for the US right based on the existing data. So in summary, we have a very differentiated and unique platform for modifying post-productions AAV with a clear demonstrations in rodent, in primate, in the retina. I didn't show data today, but in the retina and in the brain, of improved efficacy and also improved safety profile. 3 ongoing programs in preclinical using this technology and 3 ongoing partnerships. We are also very interested to collaborate with companies interested to use our technology to modify their existing AAV-based product. Thank you for your time and attention. I'm happy to take questions, if any.